one of the most important part of plant system because it is the food manufacturing site. It converts the sunlight energy into chemical energy which the plant use as food. These are the prime lateral appendage of the stem in the shoot system. They are often flat so that they absorb maximum sunlight and thin so that the sunlight can get to the chloroplasts in the cells. Most leaves have stomata which open and close. They regulate carbon dioxide, oxygen and exchange of water vapor with the atmosphere. Leaves occur in various types according to size, shape, color, texture, form and other characters. Leaf is bound by epidermal cells both on top and bottom side called as upper and lower epidermis. Anatomically, the extensive air space system, abundance of chloroplasts in the ground tissue and a close spatial relation between the vascular and ground tissue, the mesophyll, favor the exposure of the chloroplasts to light and ready access to water and gases to the cells concerned in photosynthesis. Unlike stem, the leaf commonly lacks storage tissue. It develops no periderm and majorly consists of primary tissues. No secondary growth occurs in leaf, so the leaf does not possess the capacity to restore its tissues. In perennial plants, the new leaves are constantly formed and the old leaves are shed. In dicot plants, the leaves are dorsiventral. The leaf is oriented at an angle to the main axis and held perpendicular to the direction of sunlight. It has both dorsal and ventral side with identifiable features. They are made up of three well-defined parts: epidermis, mesophyll tissue and vascular tissue. Epidermis. Epidermis is the outermost covering or boundary layer of the lamina. The leaf flat surface is overall covered by epidermal cells. The cells which cover the adaxial, that is the upper expanded surface, constitute the upper epidermis, and the abaxial, that is the underneath expanded surface of leaf, is constituted by lower epidermis. Both the upper and lower layer of epidermis consist of a single layer of cells. The cells are closely arranged without intercellular spaces except where the stomata are present. The cells are living, contain a little protoplasm with a nucleus each. No chloroplasts are present in the epidermal cells except in the guard cells of the stomata. However, in shade and aquatic plants, chloroplasts may be present even in the epidermal cells. The outer wall of the epidermal cells are thick and covered by thin or thick deposition of cutin, which in xerophytes form a thick cuticle to check transpiration. The stomata can be distributed all over the surface of the leaf. Depending on nature and the habitat of the plant system, their mode of distribution varies. In terrestrial plants, they occur more in lower epidermis. This condition is called hypostomic, whereas in aquatic plants bearing floating leaves, they are found in upper surface. In xerophytes, the ordinary epidermal cells overlap the guard cells so that the stomata are not exposed and lay a level below the epidermis. In xerophytes like nerium, the stomata occur in pits covered by epidermal hairs. This helps in checking transpiration. On the other hand, in shade plants or the plants growing in moist habitat, the stomata are raised above the surrounding epidermal cells. Stomata. Each stoma is an intercellular passage or pore bound by a pair of curved bean-shaped cells called as guard cells, and the pore is called as stomatal pore. The stomata in the lower epidermis of a leaf lead into a large irregular chamber called as air chamber, which aids in exchange of gases. Hydathode. Other than stomata, there are openings in the epidermis which are not guarded, which often appear as incompletely differentiated stomata lacking the mechanism of opening and closing. These are called hydathodes. Each hydathode may have one or more than one pore. These hydathodes eliminate water directly from the terminal tracheids. This discharge of water from the interior of leaf is called as guttation and is observed only when there is high humidity, closed stomata or active growth in plants. Epidermal outgrowths. Other than pores and openings, there occur epidermal outgrowths arising from the surface of the leaf. These are commonly the hair or trichomes. These may be unicellular or multicellular and branched or unbranched. The hair usually functions in reduction of transpiration and also sometimes produce irritating substances to discourage herbivores. Function of epidermis. The main function of epidermis is protection of inner soft tissues. It protects against desiccation of the mesophyll, and with the help of cuticle, it prevents transpiration considerably. It also provides support to the palisade tissue which hangs from the epidermis. The epidermis, with the help of stomata, aids in exchange of gases for photosynthesis and respiration. The hydathodes in epidermis remove water in guttation. Mesophyll. Meso means middle and phyllon means leaf. It is the middle part of the leaf that spreads between the upper and lower epidermis, consisting of chlorophyllous mesophyll parenchymatous tissue excluding the veins. The mesophyll is a living tissue that contains chloroplasts. The mesophyll is multilayered and is differentiated into two kinds of tissues, the palisade tissue and the spongy parenchyma. Palisade tissue. Palus means stack. The palisade tissue is so called because the cells are arranged like a row of stacks. The cells are columnar, cylindrical, arranged with their long axis parallel to one another at right angles, just below the upper epidermis in dicot leaf. The palisade is a chlorophyllous tissue and the cells contain a large number of chloroplasts as compared to the spongy mesophyll. The chloroplasts assume various shapes and position in the palisade tissue in response to the direction and intensity of light falling on leaf. Functions. The palisade is the most specialized tissue situated on the ventral side of leaf which receives the sunlight directly. Hence, it is the principal seat of photosynthesis. Spongy tissue. The spongy tissue is the other part of mesophyll situated below the epidermis. They show a wide variety of organization. The cells are irregular in shape and making an irregular network of filaments around bundle sheath, stomatal regions to enclose the large air chambers, that is the respiratory cavity. The cells of the spongy tissue contain chloroplasts, not so numerous as the cells of the palisade tissue. The characteristic feature of this tissue is that it encloses numerous large intercellular spaces that contain air, hence named as spongy or a ventilating tissue. Many dicot leaves have sclerids within the mesophyll tissue. In some secretory structures as resin ducts, lysigenous cavities and leticifers are also found. Function. The main function of the spongy tissue is to facilitate free circulation of air in the interior of the leaf and supply carbon dioxide to the palisade tissue for photosynthesis. However, it also performs the function of photosynthesis due to chloroplasts present in them. Vascular tissue. In the leaves, the vascular tissue forms an important element and is connected to the stele of the stem. In dicot leaves, the vascular supply consists of 1, 3 or many leaf traces that enter the lamina and arrange themselves in a characteristic way called as venation. The dicot leaves possess reticulate venation, where the veins form a network with the smaller bundles diverging from the larger. The larger bundles are embedded in parenchyma and are somewhat separated from the mesophyll proper. The smaller ones on the contrary form a network among the larger veins and occur in the median part of the mesophyll, usually beneath the palisade cells and in the upper part of the spongy tissue. The branching of smaller veins subdivide the mesophyll into a series of small polygons with the ultimate branches, the bundle or vein endings terminating freely in the mesophyll. As the bundles branch and become smaller gradually, the secondary tissues are reduced in amount, with xylem containing only a spiral element and the phloem of a single elongate parenchyma cell. The vascular bundles are conjoint, collateral and open. They are made up of xylem and phloem, where xylem face the upper surface and phloem face the lower surface of the leaf. The bundles are enclosed by bundle sheaths. The sheath consists of compactly arranged parenchyma surrounding the vasculature and is a channel between the vasculature and the mesophyll cells. The bundle sheath of the midrib vein is connected to the upper and the lower epidermal layers by many layers of collenchyma cells representing bundle sheath extensions or hypodermal collenchyma. The bundle sheath also conducts the flow of water from the xylem to the mesophyll cells and then to the intercellular spaces. In C4 plants, the bundle sheath cells along with the conduction are assigned to a very specific role in photosynthetic carbon dioxide fixation, in which they form a specialized compartment in which carbon dioxide can be concentrated around riboscope, thus suppressing photorespiration. The xylem is composed of various kinds of vessels, tracheids, phloem fibers and parenchyma cells. Xylem not only helps in conduction of water and mineral nutrients, but also provide mechanical support to the leaves. The phloem is composed of seed tubes, companion cells and phloem parenchyma cells. Phloem helps in translocation of prepared food materials from the mesophyll cells to different parts of the plant. In monocot plants, the leaves are isobilateral, which usually orient parallelly to the main axis and are held parallel to the direction of the sunlight. It has both dorsal and ventral side with similar anatomical arrangements. They are made up of 3 well-defined parts: epidermis, mesophyll tissue and vascular tissue. Epidermis. The epidermis is generally uniseriate as in Zea mays and Bambusa, but sometimes multiseriate as found in date palm, Phoenix sylvestris, etc. It is composed of more or less oval cells with cuticle on outer walls. The upper epidermis possess some specialized large lens-shaped elongated cells called as motor cells or bulliform cells. These cells, by change in their turgor, allow the leaf blade to curl or uncurl, a phenomenon that is advantageous to plant in restricting the loss of water from the leaf under arid conditions. Silica deposition is characteristic feature found in grass family leaves. The stomata are evenly distributed on both the surface of epidermal layer. Stomata typical of Gramineae type with noticeable air chambers, the substomatal chambers, are present in both the epidermal layers. The epidermis also possess unicellular sharp-pointed hair. Mesophyll. The isobilateral leaf mesophyll cells do not show differentiation into pellicid and spongy parenchyma, and thus the tissue are composed of mainly layers of isodiametric cells with intercellular spaces, mostly of spongy type. Chloroplasts are abundantly present in these cells. Vascular tissue. Vascular bundles are collateral and closed type as found in dicot leaf, but here they are arranged in parallel series as parallel variation is found in monocot leaf except in trillium. The vascular bundles are of monomorphic, that is same size, or dimorphic, that is smaller and larger size in nature. When dimorphic, the smaller vascular bundles are more in number. The oldest and the largest vascular bundle is found in the center. It is known as midrib vein. Generally, the vascular bundle contains xylem on the upper side and phloem on the lower side. In the xylem, only 2 protoxylem and 2 metaxylem vessels are present. The phloem consists of sieve tubes and companion cells. The vascular tissue xylem and phloem is surrounded all over by parenchymatous bundle sheath. Its cells contain plastids with starch grains. The bundle sheath of the midrib vein is connected to the upper and lower epidermal layers by sclerenchyma cells representing bundle sheath extensions or hypodermal sclerenchyma. Cran's anatomy. Plants not only modify structural or anatomical aspects of leaves but also change the functional aspect according to their requirement. The best example of anatomical and functional modification is seen in leaves of C4 plants known as Cran's anatomy. Cran's anatomy is seen in leaves of higher dicot and monocot plants. Crans, a German term, means wreath or garland. Cran's anatomy is generally associated with plants showing C4 pathway for photosynthesis. In these plants, vascular tissue system is surrounded by 2 types of photosynthetic cells, the bundle sheath cells and the mesophyll cells. Bundle sheath cells surrounding the vascular bundles are large, green, thick-walled and act as intermediate between vascular bundle and mesophyll cells. There are fewer numbers of green mesophyll cells, often extended radially from the bundle sheath. Several plasmodesmata connections are present between mesophyll and bundle sheath cells. The bundle sheath cells are unique because chloroplasts are centrifugally arranged which have unstacked thylakoid or lax grana, and the stoma is rich in starch granules. On the other hand, mesophyll cells possess randomly arranged chloroplasts which are generally devoid of starch in stroma, and thylakoid are stacked, that means well developed grana is present. Haberland called this type of arrangement as Cran's leaf anatomy. The special arrangement of cells with their respective enzymes in Cran's anatomy makes the C4 pathway very efficient and successful over the C3 pathway. Monocot leaf. The symmetry in monocot leaf is isobilateral, whereas the symmetry in dicot leaf is dorsiventral. In monocots, the stomata are distributed on both the surface equally, that is, the condition is amphistomatic, whereas in dicots, the condition is hypostomic, where the stomata is present on lower surface of leaf. In monocot leaf, the bulliform cells are present on upper epidermis, whereas in dicot leaf, they are usually absent. Only spongy parenchyma is present in monocot leaf with very small intercellular spaces, whereas in dicot leaf, both type of tissues is present, that is pellicid parenchyma and spongy parenchyma with large intercellular spaces. In monocot leaf, the bundle sheath is made up of parenchyma, but just above and below the vascular bundles, sclerenchymatous cells are found. Whereas in dicot leaf, bundle sheath is made up of parenchyma, but just above and below the vascular bundle, parenchymatous cells or collenchymatous cells are present up to the epidermis. On the basis of anatomical structure, two types of leaves have been recognized in angiosperms, dorsiventral and isobilateral leaves. The dorsiventral leaves are mostly found in Dicotyledons group of angiosperms and grow in horizontal direction with distinct upper and lower surfaces. Isobilateral types of leaves are commonly found in the Monocotyledons with no variation in mesophyll types as seen in dicot leaf. The reticulate and parallel venation in dicot and monocot respectively carries anatomical differences in their vascular system too. Plants not only modify structural or anatomical aspects of leaves but also change the functional aspect or both. The best example of anatomical and functional modification is seen in adaptation of leaves of C4 plants known as Cran's anatomy that provide some evolutionary advantage over C3 plants.